If you're interested in, let's say, human diseases, look for a lab that studies the mechanism of human diseases. Get a strong background as a graduate student, and then you can study more complex problems.
Hello everyone, welcome back for another interview in the series of interviews conducted by the Department of Chemistry, University of Colombo. Here we interview eminent scientists who have made an enormous contribution to science with the aim of inspiring youth in science. I'm Nirmali Sirivardhana and here is my colleague Pasimbu Vichayaratna and we are going to be the interviewers for today. We both are fourth year undergraduates following honors degrees in biochemistry and molecular biology. Today, I'm deeply honored to introduce Professor Lynne Maquat to our interview. To introduce her, she is a biochemist and molecular biologist whose research focuses on the cellular mechanisms of human disease. She is an elected member of the American Academy of Art and Sciences, the National Academy of Sciences, and the National Academy of Medicine. She currently holds the J. Lowell Orbison Endowed Chair and is a professor of biochemistry and biophysics, pediatrics, and oncology at the University of Rochester Medical Center. Professor Maquat is also founding director of the Center for RNA Biology and founding chair of Graduate Women in Science at the University of Rochester. Also, she is a recipient of many prestigious awards, including the 2021 Warren Alford Foundation Prize and 2021 the Wolf Prize in Medicine. The discovery of nonsense-mediated mRNA decay in human diseases, exon junction complex, stophan-mediated mRNA decay, and defining a new mechanism for microRNA's degradation are some of the immense contributions of Professor Maquat to science. Over to you, Pasimbu.
Thank you, Nirmali. Good evening, Professor Maquat, and we warmly welcome you to this interview and thank you very much for giving us your valuable time out of your hectic schedule. And to begin with, we would like to know a brief introduction about yourself, your early education, and the starting point that directed you to select the path of science and what inspired you to become a scientist.
Well, it's a pleasure for me to be here and to meet both of you. I feel honored to be asked to have an interview with you. Well, I grew up in Connecticut in the United States, which is on the East Coast of the United States. I grew up in the country, actually on my grandfather's farm. I am the first person in my family to go to college. And I was interested in a lot of things when I was a high school student. I liked music. I was fluent in Spanish. I didn't like the sciences all that much because high school science for me wasn't all that inspiring. I liked math. And I remember I got in high school the English award and the Spanish award. So you're not seeing any science in there. Even though I didn't do badly, I did very well. It didn't resonate with me. So I went to a state school. I went to the University of Connecticut when I went to college. And I took a very broad program because I wasn't quite sure what I was interested in. But in my sophomore year, when I took cell biology, and cell biology had a laboratory associated with it, and I learned about protein synthesis. I learned about what was known back then about the mechanism of how proteins are made from messenger RNAs, from mRNAs. I got really excited. I thought that it was really cool. And so I don't know how I knew to go up to the professor and ask if I could work in his lab. Actually, I asked, can I work in a lab? Because I was very shy and nervous. And he said, a lab? And so I had to tell him very nervously I wanted to work in his lab. And he said, sure. And so I started to work on messenger RNA and how it makes protein in embryonic chick leg muscle. And I found working in a laboratory it was very intimidating for me. I realized how much I didn't know, but I still liked it. And part of the reason why I liked it is because I found it was challenging. So I think I'm the kind of person who likes to solve puzzles. So that's how I got interested in science. And I learned about graduate school, and I realized I could go to graduate school not only for free, but they would pay me. At the time I got $4,000 a year, and I thought that was amazing. And so I went to graduate school at the University of Wisconsin. I was very shy, and my advisor at the University of Connecticut wouldn't let me apply to schools that were maybe more difficult. Although I have to say the biochemistry program at the University of Wisconsin was very difficult for me. And, I ended up in a laboratory that I initially didn't want to be in to do my PhD thesis work because it worked on bacteria, E. coli. And I thought I was taking a step backwards, but actually it was an excellent lab. And I learned the genetics and molecular biology of bacteria. So then when I did my postdoctoral studies, I could focus on what I really, really wanted to do, and that is study human diseases.
That's very interesting, Professor, how you became a scientist even without having a proper scientific background from your family. And it's very interesting how you became a scientist also. And Nirmali, over to you.
It's very interesting to hear this story, Professor. Next question is, you are well known for your efforts to promote women in science. So what do you think about the involvement of women in science and how they should be encouraged?
I think it is very important to have women in science. And when I was a graduate student, there weren't any female faculty members in my department, in the biochemistry department. And there were some people who didn't feel that there was a place for women in science. I know what that feeling is like, and times have changed. They're much better, but still, I think women are faced with different issues relative to men. And so besides realizing that women can contribute to science, 50% of the brain power that rightly should be doing science, and diversity in the sense that, let's face it, women are different than men. We think differently than men. We also need to nurture women because when they're going through scientific training is often the time when they're having kids, right? And so still in the United States, although it's much better, childcare falls largely on the responsibility of the woman. And this needs to be compensated for. Training grants now from the National Institutes of Health are offering, it's not a lot of money, but they're offering some money for either a male or a female graduate student who is a NIH trainee to help support childcare, to offer funding for childcare. And that's a step in the right direction. Having childcare associated with universities is a step in the right direction. University of Rochester has that. So I'm a strong proponent for women in science, and I think it's important to talk about the hurdles that might be there, both because of society and also practical considerations like how do you balance having a career with having kids? So, I mean, I think that how do we encourage women to be scientists? I think it's the way we encourage everybody to be a scientist, and that is when they're kids. And all throughout their career, help them identify as a scientist. And as soon as someone shows interest in the sciences, help them get engaged in activities so they can become educated more about what being a scientist involves.
Yes, Professor, I appreciate your contribution to encourage women in science, and it's a very advantageous way to us to encourage ourselves in science. Over to you, Asindu.
Thank you, Dimali. Let's move to the next question. Professor, your research mainly focuses on the molecular basis of the human diseases. And what made you interested in this research area specifically? And can we have a brief idea about your area of research work?
Sure. When I was a postdoc, as I told you, I was interested in studying human diseases and actually was able to show way back in 1980 that a disease could be due to a problem with pre-mRNA splicing. And these are the beta plus thalassemias, where the processing of the beta globin precursor to mRNA is inefficient. You get some normal beta globin mRNA produced in the bone marrows of beta plus thalassemic patients, but it's at abnormally low level. So normal mRNA, but abnormally low in the level of its production. And then the beta zero thalassemias, by studying those, again, using patient bone marrow samples, I was able to demonstrate that mRNA can be destabilized. And it was an actual study of mRNA half-life. And so that was my introduction into studying since then, that was 1981, the molecular basis of mRNA instability. Sequencing the alleles demonstrated that the cause was either a frame shift or a nonsense mutation that resulted in the premature termination of mRNA translation. And then we studied other diseases. We had to clone the genes way back then. The normal gene first, then the disease gene. In lambdaphage, it was a long time ago. And realized there were all kinds of pseudogenes for protein encoding genes. So we had to figure that out. And then study what happens in the patient, in the case of the patient with a gene that harbors either a frame shift or a nonsense mutation. And we again saw the mRNA was degraded, nonsense-mediated mRNA decay. And being a mechanistic biochemist, we wanted to understand, how does the cell know when an mRNA prematurely terminates translation? How does the cell know and then degrade it? How does the cell differentiate a premature translation termination codon from a normal translation termination codon, which generally doesn't destabilize an mRNA? And so we inserted premature termination codons throughout different alleles. We were working in reporters now, so we could translate introduce genes that harbor different mutations into cells in culture that were really easy to culture, like HeLa cells. And we moved the positions of introns around and realized that cells differentiated termination codons that trigger NMD, nonsense-mediated mRNA decay, from termination codons that don't, by the position of the intron within the pre-mRNA from which the mRNA derives. And the response of people was, how can that possibly be? The introns are gone from the mRNA. And I also shown that NMD required translation. And I knew that NMD was restricted to newly synthesized RNA, and it maintained an association with the nucleus in which it was made. So we did these very careful cell fractionations where we isolated nuclei free of cytoplasmic tags, but the nuclear pores were intact. And all of this was hard for people to believe. But the simplest interpretation in my mind was that pre-mRNA splicing must deposit a mark on newly synthesized mRNA that persists until the first round of translation. And then we were able to show with Melissa Moore, the mark was the exon junction complex. I proposed the concept of the mark to her. And then we defined what a pioneer round of translation is. It's using newly synthesized mRNA that people never thought could be translated. They didn't even know it was exported from the nucleus to the cytoplasm. And so, I mean, you can kind of tell that we followed our nose and thought of the simplest explanation without over-interpreting the data. And so it was a remarkable series of findings, and that we were able to show actually did occur by collaborating with Rob Singer in 2014 and looking at molecules, single molecules of mRNA using a very high power microscope. So yeah, it was a process. And of course, many graduate students, many postdocs, and many technicians who trained with me in my lab contributed to all of this work.
Thank you, Professor. That is a very interesting approach for research. And Nirmali, over to you.
Professor, your research work seems very interesting. Do you have any advice for those who want to get into your area of research?
Well, I guess my advice to get into my area of research would be the same as my advice to get into any area of research. And it depends upon what that area is and where you want to train. I would advise going to the best possible lab to study something, at least a lab that supports the graduate students and the postdocs in the lab. You can see where graduate students and postdocs go after they leave the lab. Do they go to good places? I firmly believe in understanding the mechanism of a process. I'm not a fan of correlative biology. And what I mean by that is, if something happens in this cancer cell, then I can say this happens in cancer. But that's correlative. I want to know how it happens. And I think that's the stronger science, the mechanistic biochemistry of science. And this can use many different approaches. It can use biochemistry, cell biology, sequencing, computational biology, structural biology. And I'm fortunate in my lab that I have postdocs who have come to train with me with different backgrounds. And those backgrounds include crystallography. So one postdoc is a crystallographer. I'm not a crystallographer. And I remember when the first person who was a card-carrying crystallographer wanted to train with me, I thought, oh my gosh, I'm not a crystallographer. How am I ever gonna, because he wanted to study our problems using crystallography. And it worked out fine. And now he runs his own lab. So that worked out, and he published some very nice papers. But what I'm saying is techniques are just techniques. They're very powerful. Usually a high-profile paper has to use many approaches to understand a problem. So if you're interested in, let's say, human diseases, look for a lab that studies the mechanism of human diseases. Get a strong background as a graduate student in one of they're called basic sciences. I call them discovery sciences. And then you can study more complex problems. There are a lot of complicated problems out there. But I think going for mechanism is important no matter what you want to study. So I didn't really answer your question.
Yes, Professor, I got it very clearly. Thank you for your advice, Professor. Over to you, Pasindu.
Attention, Nirmali. Professor, you have received the Canada Gairdner International Award in 2015 for discovering the mechanism that destroys the mutant mRNA in the human cells, which we call as the nonsense-mediated mRNA decay. So we would like to know the impact of this in the field of science and in medicine.
Okay. Well, when we realized that splicing of a pre-mRNA influenced, and that's happening in the nucleus, influences whether or not the mRNA undergoes nonsense-mediated mRNA decay in the cytoplasm, we figured out a rule for which premature termination codons trigger NMD and which don't, because some premature termination codons don't trigger NMD. And this rule, we call it the 50 to 55 nucleotides rule, and I won't go through the details, helps researchers and clinicians predict how the gene of interest, whether it be from a patient or something that an investigator has generated, but how their mRNA will be metabolized, whether it will be targeted for NMD or not. This rule is also helpful if you want to knock out a gene using CRISPR-Cas9 so that you make the gene non-functional. It's good to know this rule. And NMD does more than serve as a quality control pathway. I call it a quality control pathway. It is called a quality control pathway because without it, cells would produce truncated proteins that could be toxic. And in fact, those PTCs, those premature termination codons that don't follow the 50 to 55 nucleotides rule and don't trigger NMD can result in dominantly inherited disorders because the mRNA isn't reduced in abundance by NMD. The protein may not be reduced, but it's truncated. And so it can glom up the complex of proteins it normally functions in to cause a dominant negative effect in a dominantly inherited disease. So that's part of how I can answer your question.
That's very cool, Professor, how your research impact on this field of science and in medicine. Nirmali, over to you.
Professor, you're known for RNA biology in human diseases, and you have made many discoveries. So we'd like to know your most surprising discovery and how you came up with that idea. And would you mind sharing that experience with us?
Well, I think I've surprised myself a lot with the discoveries that we've made, which has always kept it interesting. The splicing-dependent mark, the exon junction complex, the pioneer round of translation, Stauffen-mediated mRNA decay. So how did we discover Stauffen-mediated mRNA decay? We were looking for new NMD factors. I mean, nothing is other than logic. So a postdoc in the lab used a yeast-2 hybrid assay. What are other human proteins that interact with the key NMD factor, UPF1? And he got Stauffen. It's a double-stranded RNA binding protein. And so, wait, if Stauffen can recruit UPF1 to mRNAs, and UPF1 is the key NMD factor, and we knew how it functioned in NMD, we knew the choreography of proteins during the pioneer round of translation and NMD. What if Stauffen recruited UPF1 downstream of a normal termination codon? That would trigger mRNA decay. So we tested that, and it does. And we collaborated with a colleague in Canada because he had already characterized 13 mRNAs that he knew would bind Stauffen in the 3' translator region, meaning downstream of the normal termination codon. And then I think the most remarkable finding recently is that nonsense-mediated mRNA decay is hyperactivated in Fragile X syndrome. Fragile X syndrome is the most common single gene cause of intellectual disability and autism. So how do we figure that out? Well, so again, we were looking for new NMD factors. And so another postdoc in the lab performed a mass spec analysis of proteins that bind to UPF1. They co-immunoprecipitate with UPF1 from human cells in a way that is resistant to RNA treatment. So in other words, you can have proteins that co-IP if they're binding to different parts of the same mRNA, but if you get rid of the mRNA, then the only way they're going to interact is if they form a complex with one another. And one of the proteins, in fact, a very high level hit that the postdoc got was FMRP. Well, wait, that's the protein that's missing in Fragile X syndrome. And so we figured out the mechanism by which NMD is hyperactivated when FMRP is gone in Fragile X. So we got into neurobiology and embryonic stem cells, induced pluripotent stem cells, differentiating them to neurons, things I never thought I would do. And thank goodness we have colleagues at Rochester who, Chris Pichel is a stem cell biologist and a mouse developmental biologist. So, he taught the postdoc how to do a lot of things. And now we have a paper that's coming out on the Fragile X mouse model of, so that we can understand when during embryogenesis, during development in utero and after birth, is NMD hyperactivated, because now we can think about therapeutics. And yeah, it's hyperactivated in starting embryonic day 16 through postnatal day 24. And so what we're doing now is, mouse brain development and human brain development occur on different timelines. And so we're correlating the timelines from reading the literature and getting ready to treat with NMD inhibitors, and also using antisense oligonucleotides to downregulate an NMD factor that we know is important for brain NMD. So it's just, a series of getting a result, what does it mean, getting at the mechanism, and I'm continually surprised. We have other projects in the lab where I'm continually surprised. We have a new signal transduction pathway for NMD. I'm continually surprised, which is good. Not to say it can't be frustrating. So look, I need to be clear to everyone that not every day is, wonderful. My feeling is if you enjoy what you're doing, let's say 60% of the time, that's a win, that's good. But it can also be extremely frustrating when we don't know what to think of a certain piece of data, or we're worried, are we asking the right questions? Those kinds of things. Yeah.
Yeah, Professor, it's very amazing to hear your discoveries and how you have done those. And I feel very interesting about those. Over to you, Pasindu.
Thank you, Nirmali. Professor, when we talk about the mRNA vaccines, they were discovered in early 1960s, but they have come to the use of humans in the near past. As an example, we use the Pfizer and Moderna vaccines to treat the COVID, to control the COVID. And so as an RNA biologist, how do you comment on the contribution of these recent improvements in mRNA vaccines to the field of vaccinology?
Oh, it's an amazing breakthrough. First, knowing that mRNA can be modified, so some of the nucleotides they use can be made pseudouridines, so that the mRNA itself in the vaccine doesn't trigger an immune response is key. And all the nanoparticle technology, how do you get the mRNA into cells so that it can reach the protein synthesis machinery, make protein, and that protein be accessible to our immune cells to raise antibodies to that protein, the viral protein, the spike protein. These are amazing breakthroughs that happened over decades. I mean, many of us decades ago tried to express an mRNA in cultured human cells. I did, so many people did. And what people have done since then is just fabulous. And of course, it's good for all of us RNA biologists, because now when I see someone on an airplane, although I haven't been on an airplane since COVID, but I'm going on Saturday, and people ask what I do, and I talk about mRNA, I'm not going to get this, but people are going to actually have something to say because they're going to understand what it is from the vaccine perspective. So it's a bad, bad pandemic, but it has put mRNA in the spotlight of the whole world.
That's a very revolutionary step in the field of vaccinology, Professor. And Nirmali, over to you.
Professor, now onwards, we would like to ask for some advice regarding the different aspects of having a career in science. So as undergraduate students, we all are doing research projects in our final year, but we have fewer experiences as we are new to research work. So when we are involved in research, what skills should we improve to make it more efficient and valuable?
Well, I think what is key is knowing what you know and knowing what you don't know. So to do something creative, it's important to know what's known, and it's important to know from what type of experiment is it known. And you'd be surprised what people assume is known, but it's really not. Or the conditions under which it's known are limiting in terms of how you need it to be known. And things are different now. There's a lot of kits, for example. It's important to know what the kit does, a biochemical kit, like I'm going to make cDNA using a kit. Well, I think it's important to know how to make cDNA, copy DNA. I think it's important for you to know what you're doing when you use a kit, and then you'll be able to troubleshoot. So no matter what you're doing, I think it's important to also keep track of how you've performed your experiments. So students and postdocs have been known to come to me with result number A and result number B. Well, those aren't numbers, but and they're very different results. And they say to me, but I did exactly the same thing. And we all know that can't be true. Keep a good notebook. I always write out experiments in advance. Then I get to think about it. Did I think of everything? Write it through all the way from step 1 to 10. If there's 10 steps, get to 4. Step 4 and realize, oh my gosh, I can't go to step 8 because step 4 needs to be altered. So I think it's really important to keep good notes, keep track of what you add to your tubes, look at your samples. Be sure when you're delivering a microliter of something, you don't have a glob of some enzyme in glycerol that's viscous at the end of your pipette man. and it's really not a microliter, but it's, 5 microliters. Just pay attention to what you're doing and ask important questions. It's easy to do experiments. It's hard to do experiments that answer a question you want to address. So have positive and negative controls. These are all logical things, but they may not pop into one's mind. And I highly recommend writing out experiments before you do them. And if you do a procedure many times, like you'll do it this week and you do it next week and the following week, you can write it out, type it out once, and you can take a blue pen and check off every step for this week. And then when you do it next week, you can take a red pen and check off every step next week. And that way, you've done what you're supposed to do. You have to pay attention. I don't think people should be listening with something in their ear, because I worry that they're not paying attention to their experiment. But I'm very old, so maybe that helps you take out background noise so you can focus even more. I'm talking about headsets and listening to music or whatever people listen to. Do you do that? Do people in Sri Lanka listen to music while they do experiments?
No, Professor, we are not usually doing that.
Okay, that's good, because they do it in the United States, and I think, it's probably not a good idea.
Yeah, sure, Professor. I got it very clearly. Thank you for your nice clarification. Over to you, Pasindu.
Professor, as an undergraduate student, how do we select a specific path of science to carry out more research in the future? And what advice would you give to us when choosing a particular area of science?
Well, I think you have to be pragmatic. And it depends upon where you can train and who is doing what science there. You want to get into a quality lab that studies mechanism, in my view, for a PhD, so that you really understand what the problems are and can figure out why you're observing what you observe. People may disagree with me, but I think it's good. It serves me well to get a really strong background in biochemistry. It doesn't matter what the biochemistry is, because it teaches you how to think. Once you know how to think, and once you know how to design experiments, then you can study other things and you'll have those capabilities. So once you know what you know and know what you don't know, then you know how to be creative, because you know what's missing. There's no one answer to your question. You certainly want to study something that you're interested in. And you may get into a lab environment and be drawn to a certain lab that you never thought you would be interested in, and it's great for you. As I said, I didn't want to train in the lab I got my PhD in. I told the poor guy who turned out to be my PhD mentor that I didn't want to be in his lab when I got assigned to his lab, but it turned out to be the best thing, because he was supportive of women in science. Remember, there weren't any female faculty members, and he was a good scientist. So it's a matter of what's available to you. There's no one answer.
Thank you so much, Professor, for your important advices. And Nirmali, over to you.
Professor, in our lives, we face many challenges and failures. One of such is setbacks in our research work. Sometimes we are afraid of those kind of failures. So as a scientist, I think you may also have had failures in your career. And how did you overcome them? Do you have any advice for youngsters who are afraid to take a step forward due to these kind of failures, fear to those kind of failures?
I have had a lot of failures. And remember, I used to be really shy. And so I have had to overcome a lot. I didn't come from a family of scientists. I didn't come from a family who even went to college. clearly my family members were smart, but I had to figure out what college was about. And when I started to work in a lab when I was a university student, I found it extremely intimidating. So I think if you believe in what you're doing, you should continue what you're doing despite your fears. So I have always been afraid of failure, and that has served me well until I got to the point where it didn't anymore, because it can be very wearing. So I gave you tips on how you can design experiments so that you help yourself not fail in executing the experiment. And with a good mentor, you can get help in designing experiments that won't fail because you're not designing the experiment properly. Perseverance is key. So you may be asked to write a grant, like a postdoctoral fellowship, and you can say, oh, I don't know if I can do it. I don't know if I'm qualified to get it. And people say to one another, well, if you don't write it, you're certainly not going to get it. And writing is an experience in itself. Writing is extremely hard. It is really hard. So you not only have to do the science, interpret the data, and be able to tell it in a logical way. You never write a paper chronologically about how you did your experiment. First I did this and then I did that, because you get surprises along the way. But telling a story when you write your paper is very hard to do so that it's a cohesive story and that it will capture the reader's interests. It's something to learn. How do you title your work? How do you title your paper? How do you write the abstract? How do you write the introduction? You know, so a common way that students and postdocs and even faculty can get stuck and feel like they're a failure is writing up their results. Like, nothing's happening. I still can do this. Okay, I'm trying to write a paper, nothing's happening. And it can take days. So what I recommend is start with materials and methods, because you know what you did. Right? So start with that. And then start with your results. We start with figures, so organize your figures. So there were many ways and many struggles to overcome when doing science. But if you have a good mentor and you have a good group around you to help you overcome these hurdles, all of which are scary, then you have to keep going. You have to keep going. Also, balance in life is key. Balance. So you cannot be working all the time, even though my husband says I do. But really, I exercise. Exercise is a very good investment in your health. You probably meditate.
Yeah.
It's good, really good to calm yourself, get enough sleep. Getting enough sleep is so important because then your brain works better.
Yeah.
So, you know, have balance in your life. It's not an easy path, but to succeed in any field, whether it be science or architecture, or you want to be an economist and you want to succeed, it's hard. No, it's hard. And it will take perseverance.
Yeah.
Don't think too far in advance, otherwise you will psych yourself out. Don't just take it step by step. Don't think, oh my gosh, I'm going to be doing this for the next 10 years. Take it step by step and do the best you can.
Thank you for your advice, Professor. Pasindu.
Thank you so much, Professor Maquat, for your inspiring words and the excellent advice given. And it's a great honor to have an opportunity for this interview with you. And once again, thank you very much for allocating your valuable time to encourage us. And we wish you all the success in your future work. And may health and happiness be with you always. And let's wrap things up. Today, we interviewed Professor Lynne Maquat from the University of Rochester, New York. And thank you for joining and stay tuned with us. Have a nice day.
Thank you,
Thank you, you
Professor.
two.